Hi, this is a short tutorial for Bioinformatics homework number 8. A lot of human disease research studies animals rather than people, and the most common research animals are mice. Like humans, mice are mammals. They have similar genomes and physiologies as humans, and develop similar diseases such as cancer, obesity, aggressive behavior, and alcoholism. Also, as we learned in the animal transformation lectures, mouse genomes can be manipulated using gene transformation, so human genetic diseases can be induced in mice. Importantly for lab research, mice are docile little animals that are relatively cheap to maintain, and they reproduce like crazy, giving birth to half a dozen or more babies up to 10 times a year. There are several places around the world that breed and maintain genetically defined strains of mice, and your task this week is to find and buy a strain of mice that can be used to study the genetic disease you identified in homework number 7. But be forewarned, not every genetic disease has a mouse model. In some cases, a mutant mouse for the disease you have identified in homework 7 may simply not exist. If this is the case, you need to go back to the Genes and Disease database at NCBI and pick another disease to investigate. In homework number 7, I used Lesch-Nyhan syndrome as an example of a monosomic genetic disorder, and this week we'll find a mouse strain that can be used to study this disease. For this assignment, we'll use the Homologene database at NCBI. This can be accessed by going to Homology, Homologene, or by typing your disease into the search box and searching the Homologene database. The Homologene results shows homologous genes and homologous protein which are found in different species of animals. Homo sapiens are humans, and your task is to find out what pan-troglodytes are. If you scroll down to the protein alignment section, you can compare the protein sequences in different organisms. We'll compare the HPRT in Homo sapiens with that in mouse, or Mus musculus, using BLAST. The result shows the amino acid sequence alignment of the HPRT gene in humans and in mice. The middle line highlights the similarity by showing the amino acids that are shared between the two proteins. In this case, 97% of the amino acids in the mouse HPRT are identical to those in the human HPRT. Now that we know what gene needs to be manipulated in mice to develop an animal model for Lesch-Nyhan, we'll go to a different database to find a mouse strain that we can use to study in the lab. The Mouse Genome Informatics website contains several databases relevant to mouse genomics. Click on Phenotypes and Disease Models, and then click on Search for Human Disease Terms to find mouse models. Here's a listing of several human genetic diseases. We're interested in Lesch-Nyhan. There's 2 Lesch-Nyhan entries. We're interested in the Lesch-Nyhan syndrome disease, not the one with a normal HGPRT gene. This takes you to the Lesch-Nyhan page. Now click on the mouse gene name under the mouse homologs, and this tells you everything you need to know about the mouse HPRT gene. It provides a FASTA file if you're interested in the sequence, and it provides a genome browser to understand the exon-intron orientation and where that gene exists on the chromosome. We're interested in alleles and phenotypes, alleles being different mutations in the same gene. There are 423 different alleles or different mutations in the mouse HPRT gene known. Of these, 395 were made by targeted gene replacement, as we talked about in the lectures. The other types of mutant alleles available for these genes are gene-trapped, transgenic, and spontaneous. 4 mouse alleles of HPRT have been annotated as linked to human diseases. Of these, the first one, HPRT-BM3, is specifically linked to the Lesch-Nyhan syndrome. Alternatively, I could have clicked on all alleles and then scrolled until I found the Lesch-Nyhan syndrome allele. Here's all the details of this particular mutation in the HPRT gene. The name of the allele is right at the top, HPRT-BM3. The mutation details show that the allele contains a 55 kb deletion spanning the promoter and first two exons of this gene. The phenotypes show the different behaviors found in either mutant females or mutant males. You can find the mice at the International Mouse Strain Resource. This shows you can get the mice either live, as sperm, or as frozen embryos. I'll order the mouse as a frozen embryo because I don't need to use it right away, and I can store the embryo in liquid nitrogen. Later I can implant it into the uterus of a surrogate mouse mother who will then give birth to the mutant mouse. And this shows how I can price it and obtain it. Do a screen capture of this page, insert it into a page in Word, and print it out. In some cases, the pricing may not be available. In that case, take a screenshot of the furthest you could get with that mouse and attach it to your homework. That's it. Your mouse is in the mail.